Dear students, good afternoon to all, and welcome to Botany sessions. In these sessions, we are going to discuss about biological classifications of Neet level question. And what is called the classification? Then why should we need classifications? See, there are 1.7 million species of plants and animals are existed on the earth. Then how can we identify those plants and animal species? So first we want to separate the plant species into groups. Then only it can possible to identification of those species. So that is why we need classifications. So classification, it is essential for identification of plant species. So it is one of the scientific approach or procedures to classify the organisms into separate group or different group based on their similarities and dissimilarities. So based on the similarities and dissimilarities, we have to separate the group of organisms. We may easily identify it. That's why we need classification. So the scientific approach of classifications, this effort are first made by Aristotle. Aristotle, he was called as a father of biology as well as father of zoology. He is the first person to make an effort to classification of living organism in the basis of scientific effort. So he divided the whole living organism into two. One is plants, the other one is animal. So the plants are further classified into herbs, shrubs, then trees. See, it is one of the most simplest classification based on their morphological characters alone, or based on their external features alone. He classified the plant into herbaceous plant, shrub, and trees. Herbaceous is nothing but a soft plant. A large size of a soft plant is called shrub. A large size herb is called trees. So it is a simplest type of classification. And he divided the animal into two groups. One is Anaima and Anaima. Anaima and Anaima. Anaima is nothing but the animal which have, without RBC. Animal without RBC, red blood corpuscles. So those organisms are animal with RBC, having RBC. These are called Anaima. With the RBC, Anaima. Without RBC, Anaima. So this is also one of the simplest classifications was done by Aristotle into two plants and animals. Then followed by Carolus Linnaeus. He proposed two kingdoms of classifications. Carolus Linnaeus, he was a Sweden botanist to classify the living organism into two kingdoms. One is Plantae, then another one is Animalia. Plantae and Animalia. So he divided the whole organism into Plantae and Animalia. So he is called as a father of taxonomy, Carolus Linnaeus, and he divided the whole kingdom into whole organism into Plantae and Animalia. But certain defects and defects also there in the system of classification. In this system of classification, does not distinguish or not differentiated into unicellular, not differentiated into unicellular organism and multicellular. Then prokaryotic organism and eukaryotic organism. Eukaryotes. Then photosynthetic and non-photosynthetic organism. Non-photosynthetic organism. So in this system of classification, not differentiate the unicellular organism and the multicellular organism, prokaryotes and eukaryotes, photosynthetic organism and non-photosynthetic organism. All these organisms are kept under these two kingdoms, Plantae and Animalia. That is a defect and a demerits of in this system of classification. That is why to evolve three kingdoms system of classifications. So the three kingdoms system of classifications was proposed by Ernest Haeckel. Three kingdom are proposed by Ernest Haeckel. So he suggested the third kingdom. It is nothing but Protista. It is the third kingdom suggested by Ernest Haeckel. The Protista, it includes all unicellular and eukaryotic organisms. Unicellular, the entire body is made up of only one cell. A single cell can carry out all the life processes. Eukaryotes, the word "eu" means two. Karyo means nucleus. That means two nucleus is present. So those organisms have a true nucleus and a unicellular. He kept under the group is called or kingdom is called the Protista. So what are the three kingdom? One is Protista. Then second one is Plantae. Second is a Plantae. Third one is Animalia. So these are the three kingdoms were proposed by Ernest Haeckel. All eukaryotes. For example of eukaryotes, unicellular eukaryotes, fungi, bacteria, protozoans, algae, then slime mold, the next bacteria. So these are the organisms which kept under the group. It is nothing but Protista, fungi, bacteria, protozoan, algae, and slime molds. But here, what are the defects or demerits of in this system of classification? Bacteria, it is primitive organisms. Here, the true nucleus is absent. It is comes in the category of prokaryotes, but he kept it. Ernest Haeckel kept it in the eukaryotes. So there is a drawback or demerits of in this system of classifications. So that is why the four kingdom system of classification is evolved. So these four kingdom. Yes. The four kingdom system of classifications were proposed by Copeland. Four kingdom system of classifications were proposed by Copeland. So he suggests the fourth kingdom. It is nothing but Monera. The Monera, which includes all prokaryotes. all prokaryotes and unicellular organism. Unicellular organism. So all the unicellular and prokaryotic organism are kept under the kingdom Monera. So the group Monera was established by Copeland. So what are the four kingdoms? First one is Monera, the second Protista, then third one is Plantae, then fourth one is Animalia. So these are the four kingdoms system of classifications proposed by Copeland. But what are the defect and demerits of in this system of classification? Certain non-photosynthetic organisms also kept under these kingdoms. These are demerits or drawback. That is why we evolved a 5 kingdom system of classifications. So the 5 kingdom system of classifications are proposed by R.H. Whittaker. R.H. Whittaker. In the year 1969, R.H. Whittaker proposed 5 kingdom system of classification. He is the first person to separate the fungi. Actually, fungi is a non-photosynthetic organism because of lack of chlorophyll pigment. They do not prepare their own food material. It comes under the category of epitrophic organism. He separated the fungi from Plantae, kingdom Plantae. He given the separate kingdom fungi. So what are the first kingdom of 5 kingdom classification? The first kingdom Monera. First one is a Monera. The second Protista. Third fungi. It's a correct order. Third one is a fungi. Fourth Plantae. Fifth one is Animalia. Animalia. So based on the Neet point of view, these 5 kingdom system of classifications were based on certain criteria. So what are the criteria to follow to classify these 5 kingdom? The first one is cell structure. Cell structure. Whether it is a prokaryotes or eukaryotes. Prokaryotic organism or eukaryotic organism is the first criteria to follow while you are classifying this 5 kingdom classification by R.H. Whittaker. Then second one is thallus organizations. Thallus organization. It is nothing but plant body or a body, whether it is differentiated or non-differentiated. So body differentiated or not differentiated. It is second criteria. The third one is mode of nutrients. Mode of nutrients. Whether it is autotrophic organism or heterotrophic organism. These are the mode of nutrients are following. The next fourth one is method of reproduction. Method of reproduction. Then fifth one is phylogenetic relationship. Phylogenetic relationship are also called evolutionary relationship. So these are the 5 criteria to following by R.H. Whittaker to classify these 5 kingdom. Among these, which one is a most widely used for classification? Mode of nutrients. The mode of nutrients are widely used for classifying these 5 kingdom system of classifications. The next, what are the defects and demerits? What are the merits of this 5 kingdom system of classification? Fungi has been given a separate position by R.H. Whittaker. It is the merits of this system of classification. But demerits, virus is not clear about these kingdoms. No one placed in any kingdom virus. So that is why the 6th kingdom also system of classifications also evolved. What are 6th kingdom? The 6th kingdom system of classifications are proposed by sixth kingdom. Yes, the sixth kingdom. They were proposed by Carl Hughes. Six kingdom classifications proposed by Carl Hughes. Carl Hughes actually he divided the Monera into two groups. Monera divided into two groups. One is Archaebacteria, and another one is Eubacteria. So he divided the Monera into two groups. First one is Archaebacteria, second one is Eubacteria, the third kingdom is Protista. Fourth one is fungi. Then fifth one is Plantae. Sixth one is Animalia. So there are the six kingdoms that are proposed by Carl Hughes. Why he separated Monera into two groups? Because the Archaebacteria is one of the most primitive bacteria. It is entirely different from the Eubacteria in the form of cell structures. Take cell structure. In Archaebacteria, cell wall is made up of a non-cellulosic polysaccharide. It is nothing but pseudomurine. But whereas in Eubacteria, cell wall is made up of peptidoglycan. So that is why the Archaebacteria differ from Eubacteria. So he divided Monera into two groups, Archaebacteria and Eubacteria. Third one is Protista, fourth one is fungi, fifth one is Plantae and Animalia. And based on their sequence of R16X rRNA, based on the sequence of 16S ribosomal RNA gene. Because the ribosome only it is a cell organ present the most primitive organism bacteria. Ribosomal RNA, based on the sequence of this ribosomal RNA, the whole organism it is grouped into three categories by Carl Hughes. The three categories commonly called as domains of life. So it includes three domains. The first one is bacteria. Second, archaea. Third, eukarya. So these are the three domains of life that are proposed by Carolus Linnaeus. So these are the six kingdom of classifications. Now let us discuss next body rocks. Let us discuss about some groups of body rocks. The body rock, which includes Eubacteria, Archaebacteria, Cyanobacteria, then mycoplasma. Take first one, Monera. What are the characteristic features of Monera? Monera, which includes are unicellular and prokaryotic organisms. Unicellular and karyotic organism. The word pro means primitive. Karyo means nucleus is the most primitive nucleus. The nucleus is not well developed. For example, bacteria. Let us see the diagram. Bacteria. Yes, it is a suture of bacteria, ultrastructure of bacteria. In the bacteria, there is a most primitive nucleus. Nuclear membrane is absent. The nucleus does not enclose by membrane. So the DNA is a natural DNA, circular natural DNA. It is nothing but incipient nucleus. Incipient nucleus are also called genopore. Genopore. So nucleus is not well developed, not enclosed by membrane. Such nucleus are called incipient nucleus, are also called genopore is present. If you take the outermost layer of this bacteria, cell wall is present, but cell wall here is made up of peptidoglycan. Cell wall, peptidoglycan. It is nothing but peptides, proteins, or amino acids. It's made up of amino acids and sugar. Glycan is nothing but sugar. Then this glycan part, it's a that form a backbone of the cell walls. The glycan part of this compound is to form a backbone of the cell wall because it's a made up of or composed of two amino sugar which is arranged alternatively. Two amino sugar arranged alternatively. Yes. The first sugar, it is nothing but N-acetyl glucosamine. Glucosamine. N-acetyl glucosamine. The alternative, another amino sugar is nothing but muramic acids. N-acetyl muramic acid. Muramic acid. So it is NAG, then NAM, N-acetyl muramic acids. But these two amino sugars are linked by 1,4 glycosidic bond. Glycosidic bond. Which is nothing. These are the sugar molecule. I mean, one carbon of the first sugar molecule and the fourth carbon of the second sugar molecules are linked by glycosidic bond. So these are the backbone of the cell wall in eubacteria. Then actually only cell organs are present. All the membrane bound cell organs also are absent in bacteria, except the ribosomes are present. The ribosomes are oftenly occurs as a polysomes or polyribosomes. We know that there is a many ribosomes attached to the single mRNA. These are polysomes. Ribosomes are the actual site of protein synthesis. So what is cell organs? The next, mitochondria is absent, but these functions of mitochondria is carried out by mesosomes. Then what is called mesosome? It is an analogous organs of mitochondria. You know what is called analogous? Structurally differ, but functionally same. So the cell membrane which fold inwarded, the infolding of cell membrane to form a sac-like structure called mesosome. The mesosome to carry out the functions of mitochondrial functions. The next, certain fat droplets also present in the cytoplasm of the cells, which is a locomotory organ of the bacteria. Flagella, it is a locomotory organ. And another small state, hollow-like structures, which arises from the cell membrane, that is called pili. It is a made of proteins called pilin. Flagella is made of proteins called flagellin. What are the functions of this pilin? There, which is involved in the sexual reproductions. How the bacteria are involved in sexual reproduction? There is no any specialized organ for sexual reproduction. But even though they transfer the genetic material from one bacteria to another bacteria through a tube, that is called conjugation tubes are formed. These conjugation tubes are formed between these two bacteria with the help of pili. So they are called sex pili. So what are the functions of pili? They are involved in the transfer of genetic material. What are the function of flagella? These are locomotory organs. In addition to that, there is a straight, short hair like how to grow the arises. It is nothing but fimbriae. Then what are the functions of fimbriae? Fimbriae, it is an organ of attachment. They attach to the substrata. So these are the three filament-like structures. One is a flagella, another one is a sex pili, the third one is a fimbriae. Remember, if you take the flagellum is composed of three parts. The base part is called basal body. Base, it is a basal body. Next, a hook-like structures from which arises filament. Yes, it is a hook-like structure, basal body, then filament-like structures. This flagella arises from the cell membrane. It is composed of four rings are present. Let us see the diagram. Four rings. Yes. It is a four rings of flagella. It is composed of four rings. The basal two ring is nothing but M ring and S ring. It is attached to the plasma membrane. The M that refers to membrane ring. It is a membrane ring. Membrane ring attached to the plasma membrane. S that refers to super membrane ring. Super membrane ring. These two rings attached to the membrane that is called the plasma membrane. The outer two rings, P ring and the L ring. The P that refers to peptidoglycan ring. It is attached to the cell wall. Peptidoglycan. Then another one is a L ring. It is nothing but lipopolysaccharide ring. Lipopolysaccharide ring. This outer two ring is called L and P ring. Inner two ring is called S and M ring. So it is a question. And there is a hook-like structure. From the hook-like structure, the filament is arises. So the four rings are present in the bacteria. Now, how these bacteria reproduce and also what are mode of nutrients? The first, let us see the what are mode of nutrients. Based on their mode of nutrients, they are divided into two. One is autotrophs and heterotrophs. The bacteria, they are autotrophic organism. They manufacture their own food material either through light or maybe chemical substance. If they use the solar energy or light energy for the synthesis of their own food material, they are called photoautotrophs. For example, by using solar energy for the synthesis of carbohydrate. For example, purple sulfur bacteria. Purple sulfur bacteria. The purple sulfur bacteria, due to the process of your green coloring pigment, is called bacteriochlorophyll pigment. Bacteriochlorophyll pigment. So, due to the process of this bacteriochlorophyll pigment, they utilize light energy to synthesize the carbohydrates. So, how they fix the carbon dioxide? The carbon dioxide is fixed by an inorganic sulfur. The inorganic sulfur to fix the carbon dioxide to release water and also plus energy also releasing. Remember, these organisms are called anorganic photosynthesis. Then why do we call anorganic photosynthesis? Oxygen is not evolved during photosynthesis because instead of that water, they fix. Instead of the water, it's use sulfur. If they use the sulfur, they want to release oxygen. So these are called anorganic photosynthesis. So here, which one is an electron donor? Sulfur is an electron donor in purple sulfur bacteria. The second one is green sulfur bacteria. Green sulfur bacteria. The green sulfur bacteria, it contains a chlorophyll pigment is called bacterioviridin pigment. Bacterioviridin pigment. Due to the presence of this pigment, they manufacture their own food material. Then which one is an electron and proton donor? Electron and the proton donor is nothing but H2S hydrogen sulfide is an electron and proton donor. So it is also called an anorganic photosynthetic organisms. So these are also called photoautotrophs. If they utilize chemical energy, those bacteria which utilize chemical energy for the synthesis of food material, these are called hemoautotrophic bacteria also. The chemical, how they obtain the chemical energy by the oxidation of various inorganic substance. Inorganic substance. For example, nitrate, ammonia, NH3 ammonia. These are the inorganic substance. By the oxidation of these various inorganic substances to release energy, this energy is replaced for synthesis of carbohydrate. Example, nitrosomonas. Then another one is hydrogenomonas. These are the example for hemoautotrophic bacteria. It's a mode of nutrition. And based on their reproduction, how they reproduce. There are two methods of reproduction, asexual and sexual method. Mode of reproduction. One is asexual and sexual method. The asexual method, they do not involve diffusion of gametes, but certain asexual spores are formed. For example, idea, conidia, sporangiosperms, and these are the certain asexual spores. We then form these asexual spores to form new bacteria. These asexual spores. And one of the most common method of asexual reproduction is called binary fissions. So within a fraction of second, the bacteria divided into millions by direct cell division. It is the binary fission. The single bacteria divided into two equal halves, binary fissions. And in addition to that, endospore formation. During unfavorable conditions, so bacteria, during unfavorable conditions, non-availability of food material, they undergoes dehydrations, removal of water molecules. The protoplasm are get shrink. They secrete a thick wall around itself. These are called endospores. Endospores. The endospores is highly resistant to the heat. They can withstand the higher temperatures also because this thick wall containing calcium picolinic acids. Calcium picolinic acids. Due to the presence of these calcium picolinic acid, they can easily resist against some heat. When the conditions are favorable condition, it undergoes hydration, addition of water, rupture the thick walls and also release to develop another bacteria. So the asexual reproduction. If you take the sexual reproductions, there is no any specialized gamete or formed for the fusion of gamete because it's a lowermost organism, no gamete formations. But even though sexual reproduction takes place, how they reproduce sexually? How they reproduce sexually? By three methods. One is transformation. Transformation. Second one is transduction. The third one is conjugations. Conjugations. Transformations. So the transfer of genetic material from one bacteria to another bacteria, these are called transformations. It was first demonstrated by Griffith. Demonstrated by Griffith in the year 1928. Transformation by using mouse. Transductions, a transfer of genetic material such as double-stranded DNA, a pieces of double-stranded DNA from the donor bacterium to the recipient bacterium. It is carried by bacteriophage. So here the bacteriophage, which acts as a mediator to transfer of genetic material from donor bacteria to the recipient bacteria. It was first demonstrated by Letterberg and Tatum in E. coli bacteria. Letterberg and Tatum. Third one is conjugations. Transfer of genetic materials from one bacteria. For example, the bacteria which has plasmids, these are called fertility factors, as we know that. Or the bacteria which do not have plasmids, called F minus, also lack of fertility or female. So among the double strand of plasmid, single strand which is passed from one bacteria to another bacteria through conjugation tube. These are called conjugation method of reproduction. It is first observed by it's observed by Letterberg and Tatum. Transductions, Ginder and Letterberg. Remember, transduction was first demonstrated by Ginder and Letterberg, and conjugations, Letterberg and Tatum. So these are the method of reproduction in bacteria, sexual reproduction. There is no formation and fusion of gamete, even though sexual reproduction achieved by these three type of methods. It is also called a genetic transformation in eubacteria. Next, archaebacteria. Second, moving on. Archaebacteria. See, it is one of the most primitive bacteria. The simplest bacteria is called archaebacteria. The archaebacteria can withstand extreme environmental conditions, extreme or harsh habitat. These bacteria can survive in the harsh habitat or extreme environmental conditions. What is the reason? Due to the presence of a branched lipids. Due to the presence of branched chain of lipid in cell membranes. Due to the process of branched lipid chain, they decrease the fluidity. They decrease the fluidity in the membrane. If they decrease the fluidity in the membrane, definitely both the internal surface and external medium is a hypertonic solution, they can easily survive. If the fluidities are increased inside the membrane, what will happen? The fluidity is become hypotonic solutions. When compared to external medium, if the fluidity are hypotonic, definitely according to osmosis, the water molecules are move from high to low. Automatically, this bacteria cannot survive. So, how can survive? To decrease the fluidity in the cell membrane due to the presence of this branched chain of lipids in the cell membrane. That is why they can survive in the extreme or harsh environmental conditions. So, based on these three types, and the first one is methanogens. This type of bacteria can live more extreme marshy places. These methanogen bacteria have the capacity or are capable of conversions of carbon dioxide and methanol into methane. CH4 methane. See, these methanogens bacteria to convert carbon dioxide and methanol in addition to the formic acids also into methane or also biogas. The biogas is also called Bobar gas because this bacteria are present in the guts of every ruminant animals. In the ruminant animals, conversions of this carbon dioxide, methanol in the cow dung into methane or biogas productions. So that is why it's called a methanogen. Remember, methanobacteria. Methane production, biogas productions, methanogen bacteria. Example of methanogen, methanococcus. Then methanobacterium. So these are the bacteria present in the guts of every ruminant animal, for example, cow and buffalo. They act on the cow dung, which convert carbon dioxide, methanol into methane productions. So second one is halophils. It is another archaebacteria. The halophils can survive the extreme salt conditions. Salt waters. Extreme salt conditions, halophils. How they are survived extreme salt condition? Because of presence of this branched chain of lipids are present. That is why they can easily survive the extreme salt condition. Halo means salt waters. For example of these halophils, halococcus. Another one is a halobacter. So these are the two examples of halophils. The next, thermoacidophils. Third one is thermoacidophil. thermoacidophils. This type of bacteria capable of tolerance of extreme temperatures. 80 degrees Celsius can able to tolerate. In addition to that, very low pH. Very low pH. The pH is 2. This type of bacteria can tolerate the extreme or higher temperatures 80 degrees Celsius at a very low pH. Also can tolerate extreme acid. They can survive with the extreme acid. For example, they convert their oxidized sulfur. Sulfur gets oxidized to form sulfuric acids, H2SO4, sulfuric acid. They can survive with the extreme acid also. Thermoacidophils. For example of these thermoacidophils, thermoplasma. These are the bacteria example of thermoplasma. So these are the 3 types of bacteria. It's called archaebacteria. This is one of the most primitive, and also they differ from the true bacteria based on the cell wall structures. The cell wall is composed of non-cellulosic polysaccharides, are also called pseudomurin. But eubacteria cell wall is made up of peptidoglycans. It is also called mucopeptides. So second. Let us see the next. Third one is cyanobacteria. Third, cyanobacteria are also called blue-green algae. What is called cyanobacteria? It is a nitrogen fixing gram-negative photosynthetic organism. These are called cyanobacteria. Nitrogen fixing gram-negative photosynthetic organisms. But here in the cytoplasm of the cells contain 70s type of ribosomes present. It is also moneurons. All the prokaryote organisms, there are 70s type of ribosomes are present. Here also true nucleus is absent. But see the certain example. For example, of cyanobacteria, Anabina, Nostoc, Alocera, Oscillatoria, Anabina, Nostoc, Oscillatoria, Limbia. So these are the certain example of cyanobacteria. Let's see the diagram. Yes, it is a Nostoc. In Nostoc, It is a filamentous-like structures. They can able to fix the atmospheric nitrogen by a structures is called heterosis. See how they fix the atmospheric nitrogen under anaerobic conditions. In the absence of oxygens, they can able to fix the atmospheric nitrogen. For this purpose, some of the cells they become pale yellow, and the thick-walled cells are formed. This thick-walled cell is called heterosis. This heterosis cell can perform nitrogen fixation. So every cells of these filamentous of blue-green algae, Nasta the cyanobacteria, it covered with a mucilaginous substance is present. Due to the mucilaginous layer, they are said to be mixophyceans. Sorry, mixophycean. In addition to this, the cyanobacteria also they form a symbiotic association with the coralline roots of cycas. Coralline roots of cycas. For example, anabina. Anabina also it is a filamentous-like structures. Anabina. The anabina they can fix the atmospheric nitrogen with the coralline roots of cycas, gymnosperm cycas. They form a mutual relationships or mutual benefit of these two living organisms. So these are called symbiotic associations. So it is a first oxygenic photosynthesis have been reported to evolutions of oxygen during photosynthesis. Keep in mind. Remember, these are the first oxygenic organisms. The first oxygenic organism have been reported to evolve oxygen during photosynthesis. See how they perform photosynthesis due to the presence of the chlorophyll pigments. So what are the chlorophyll pigments are present? The photosynthetic pigments. Photosynthetic pigments is nothing but chlorophyll A. It is a universal pigment present in all. And next, phycobleans. The phycobleans may be phycoerythrin, phycocyanin, and allophycocyanin. These are the photosynthetic pigments that are involved in the photosynthesis. The phycoerythrin is also called a red coloring pigment is present, and the phycocyanin is also called blue coloring pigment is present. Allophycocyanin is also light blue coloring pigments. Blue color pigment is present. So as a result of photosynthesis, the photosynthetic products are stored in the form of cyanophycean starch. Cyanophycean starch is present. These are the reserve food materials in the blue-green algae or cyanobacteria. And in addition to that, gas vacuole is present. Gas vacuoles. Gas vacuole is present. Yes, it is a gas vacuole present in the cytoplasm. Instead of that, sap vacuole. There are 4 types of vacuole: gas vacuole, sap vacuole, contract vacuoles, and food vacuoles. There are 4 types of vacuole. In the cyanobacteria blue-green algae, the gas vacuole is present instead of that sap vacuole. Sap vacuole is present only in parts, but here gas vacuole is present. Due to the presence of this gas vacuole, they are easily floating or buoyancy. They help in buoyancy of the organisms. Gas vacuoles are present. So these are the structures of cyanobacteria, are also called blue-green algae. The next, mycoplasma. The fourth one is mycoplasma. It is the smallest cells like PPLO, pneumonia like organism is a smallest cell. It was first discovered by rocks and Novak. Lockhart and the rocks to discover these mycoplasma are smallest cells of organism from pleural fluids of bovine cattle. Pleural fluid of bovine cattle, African bison, bovine cattle, which is suffer from pneumonia. It is a pulmonary disease. So he find out or discovered this smallest organism from the pleural fluid of bovine cattle, which is suffer from pneumonia diseases. So these mycoplasma is a cell-wall-less organism. Let us see the diagram. See the diagram. Yes, it is a structure of mycoplasma. Yes, cell wall is absent. Cell wall absent. Cell wall absent. But only plasma membrane also presents. It is a 3 layers of plasma membrane. It's called lipoprotein membrane, 3 layers. So inside this genetic material is called DNA, but here the DNA it is a double stranded, but not in circular in manner. It is a linearly arranged. DNA also present. In addition to DNA, soluble RNA also present. Soluble RNA also present. It is a cell wall-less. Cell wall is absent, but they can survive even in the absence of oxygens. They can survive absence of absence. They can survive in the absence of oxygens. So due to the absence of cell wall, it is a pleomorphic organism. Pleomorphic. The word pleomorphic, it is nothing but pleomorphic is nothing but they often change its shape. So that is why it is called as a joker of the plant kingdom. Jokers of plant kingdom, which is a jokers of plant kingdom, mycoplasma, because they are awfully changed because cell wall is absent. So only plasma membrane is present. They awfully change its shape, so they are called jokers of the plant kingdom. And it also appears like a fried egg. Fried egg appearance in the culture medium. Like the culture in the laboratory condition appears like a fried egg. It is also cause some diseases, both plants, animals, including human beings also. Some plant diseases, for example, bunchy top of papaya. It is a plant diseases. And witches brooms of potato. Little leaf of brinjal. So these are the 3 diseases which are caused by mycoplasma. So now Manira over. Let us see the some important points about this one. Second kingdom, Protista. protista. All includes all unicellular eukaryotic organisms. Eukaryotic organism. The word true nucleus is present. All the membrane-bound organs also present. And here 80s ribosomes. 80s ribosomes are present. It's a eukaryotic organism. And prokaryotic organism only 70s type of ribosome present. Here 80s type of ribosomes are present. And cilia flagellum, it is a rhomboid organs of protista. Cilia flagella. And they are arranged in 9+2 arrangement of microtubules. The microtubules, they are organized in 9+2. I mean, if you take the cross sections of cilia flagella, there is center singlet doublet fiber. There is a periphery 9 singlet fibers. 3, 6, 7, 8, 9. Yes. These are the 9 peripheral spokes. 9 peripheral spokes centered to fibrils. So these are called 9+2 arrangement. It is a 2. 9+2 arrangement of microtubules are present in the eukaryotic organism. And all the membrane-bound cell organs also present. What are the mode of nutrients? Based on their mode of nutrients, they may be holophytic, then holozoic, absorptive types. Holophytic, photosynthetic organism. Autotrophs are called holophytic. And holozoic, ingestion types. I mean, the food metals are ingestion. The food metals are take and digestion within the body. These are called ingestional types. Ingestion. And absorptive types, certain protozoans, they get their nourishment from other living, maybe that's called parasite, or other dead and decaying organic matter. These are called saprophytic organisms. It is a mode of nutrients. Nearly 80% of photosynthesis in the earth are performed by protista. 80% of photosynthesis on the earth is performed by protista. Now, how the protists are classified? Next, the protista is divided into 3 categories. One is photosynthetic protista. It is also called the autotrophic protista. Then second one is consumer or decomposer or saprophytic protista. The third one is protozoan protista. The protozoan protista, for example, zooflagellates, protozoans, amoeba. So these are the examples for protozoan protista. Consumer decomposes are saprophytic, for example, slime mold. This slime mold, it possesses a characteristics of both plants and animal characteristics present. Initially, many of the mycologists consider a fungus, slime mold. This is an example for saprophytic protista. The photosynthetic protista. Photosynthetic are also called autotrophic protista. is further grouped into three categories. Three categories. Photosynthetic protista. The first group is called chrysophytes. Chrysophytes. Chrysophytes, it is also called basilaриophytes. By A class. Basilaриophyceae. Chrysophytes. For example, diatoms, and desmites. Diatoms and desmites. The second one is dinoflagellate. Dinoflagellate. It's contain only two flagella. Example, navicula, then gymnodium, borealex. These are the examples for dinoflagellates. And the third one is euclinoids. For example, euclina. So the three categories. Let us see the one by one. The first one is chrysophytes. It's also called photosynthetic protista. For example, diatom and also desmites. These are mostly aquatic organisms. Diatom. It is commonly called, both are commonly called as golden brown algae. Then why do you call it as a golden brown algae? Because of various photosynthetic pigments such as chlorophyll A, C, fucoxanthin, diatoxanthin, So these are various color like yellow, orange, and red color pigments. So they are said to be golden brown algae. The cell wall of these diatoms, it is indestructible. We can't easily destroy. The cell wall is not easily destroyed because due to the deposits of silica depositions. So these are said to be siliceous cell wall, silica deposits. But the cell wall of this organism of diatoms actually, it is consists of two overlapping shell. It look like a soap bar like such as two overlapping. Upper layer is called epitheca. The lower layer is called hypotheca. So this siliceous shell, is there should be pile of one above another to form a heap of diatomaceous earth is formed. Diatomaceous earth. So the diatomaceous, which is used as a cleaning agent in toothpaste and also metal polishing. It is also used for the filtrations of alcohol, antibiotic filtrations, and it is also used for making soundproof room. These are called diatomaceous earth because of due to the deposits of silica depositions. Diatomaceous earth is obtained from diatoms that belongs to chrysophytes protistans. The reserve food materials in the form of leucocytes. These are called photosynthetic producers. They produce the end product of photosynthesis stored in the form of leucocyte. These are the reserve food materials in these organisms. The next, dinoflagellate. The second one is dinoflagellate. It is also called golden brown algae. Golden brown algae. And it is composed of only two flagella. These flagella are called heterocon flagella. Let us see the diagram. Yes, it is a dinoflagellates. Each composed of two flagella, I mean two grooves. Each cell has two grooves. One is a transverse grooves. These are called transverse groove. It is a transverse groove. Another one is a longitudinal groove. It is a longitudinal groove. So due to the presence of these two grooves, the two flagellum is arises. This flagellum is called heterocont. Heterocont. In the sense, two different flagella. The flagellum which is arises from the transverse grooves or furrows, these are called smooth ribbon-like structures. The flagellum which is arises from the transverse groove is look like a ribbon-like structures. Those which one is arises from the or lies from the base or coastal regions of this transverse grooves, sorry, longitudinal grooves, these are called smooth one. This is a smooth one. So due to the presence of these two different flagellum, these are called heterocont. So both the flagellum which is arises from the right angle to each other, which making a peculiar movement, spinning movement, rotating movement, these are called whirling whip. It's called whirling whip. Whirling whip. The two flagellum, both are lies or arises from the right angle to each other, which making a peculiar spinning movement is called whirling whip. So it is also photosynthesis. They perform photosynthesis. They store the reserve food materials, reserve food in the form of carbohydrate and oils. These are the reserve food materials are present. See, these are the certain example of dinoflagellate. Every cell is composed of only two flagellum is present. And in the Neet point of view, certain example, some of the dinoflagella which cause red tide. Red tide is caused by gymnodium. Another one is Godialax, gymnodium and Godialax, which impart or give red color to the sea. So they are called red tide. So these are extensively growing in the sea, which give red color to the sea. So they are called red tide. Red tide is caused by gymnodium. The other one is some of them which cause bioluminescence. Bioluminescence is nothing but in the night time which emit light. For example, Natulaka. Is a dinoflagellate that emit light at night. That's why the seas are blue at the night times. So by the process called phosphorescence. So this is also called bioluminescence. You keep in mind, for example, Natulaka. Let us see next. The third one is Euglena. Yes. It is a Euglena. It shows both the plant and animal characters. These are the spindle shape like such as the posterior end is gradually gets reduced to like a tail like such as the tail portions, and the anterior portion is composed of three parts. One is a mouth regions, is called stomium. Other one is a canal, is called cytopharynx. It's a canal. And a basal region is called reservoirs. Reservoirs. So there is a single flagellum which is arises at the base of the reservoirs, but actually there are two flagellum is arises. Two flagellum is arises. See, the one is a short flagellum. Another one is a long flagellum. The long flagellum, which is branched into branches, into two branches. The long flagellum. So the point from which where the two branches are arises at this region, there is a occurrence of paraflagellar body. Paraflagellar body is present. Paraflagellar body is present. And there is a high spot, equal portions of equal level. There is a high spot also present, which contain a photosensitive light. Photosensitive. It is nothing but stigma. The stigma which is present on the equal level of paraflagellar body. They are responsible for perception of light. It is also called a high spot. In addition to that, what are the characteristics of animal character? Contractive vacuole is present. So, what are the functions of this contractive vacuole? They are responsible for excretion functions, nucleus. Then, what are the plant characters? Chloroplast is present. It is a chloroplast. So, the chloroplast, then the pigment which is involved in photosynthesis, which is present in the euglena, chlorophyll A, chlorophyll B, then xanthophyll. Xanthophyll is present. They are responsible for photosynthesis. Chlorophyll A, chlorophyll B, xanthophyll. As well, the reserve food materials are stored in the form of paramylon bodies. Paramylium or paramylon bodies. These are called reserve food materials. In addition to that, here cell wall is absent. So that shows is the characteristics of animal character. But the outermost layer of the organism, I mean euglena, covered with a proteinous body is called pellicle is present. These are called proteinous body. Proteinous body is called pellicle is present. Pellicle. So these are the structures of euglena. Now take break for 5 minutes, and after 5 minutes we are going to discuss some questions which are related to this topic. Okay, yes. Some questions I'm going to discuss which are related to the topic. The NCERT based questions. Let's see the questions. Which statement is wrong with respect to the kingdom protista? We know that what are the characteristic features of protista? A single-celled eukaryotes are based on the kingdom. It's correct. All the protists are unicellular and eukaryotes. Second, the boundaries of this kingdom are not well defined. Correct. That's why the boundaries. And the next, third one is chrysophytes, dinoflagellates, euclinoids, slime mold, and protozoans are included under this protista. It is also correct. And the fourth one is all protista are eukaryotic. It is correct. Then A chlorophyllus, it is wrong. All protista are photosynthetic protista. They are chlorophyllus. Heterotrophic, autotrophic mode of living is also present. It is also wrong. Non-vascular organism, yes. These are non-vascular organisms. And which one is a wrong statement with respect to the kingdom protista? Only fourth one. So the correct option is third one. See the next questions match the column. Column ones are given chrysophytes, dinoflagellates, euclinoids, slime mold. And the chrysophytes, for example, golden brown algae. First one, 2. Chrysophytes. It is also called golden brown algae. For example, diatoms and desmites. Dinoflagellates, gonialax. Dinoflagellates. Second one is gonialax. Third one is euclinoids. Euclinoids. Absence of cell wall. There's a pellicle is present. So third one. Fourth, slime mold. It is a saprophytic protista. Fourth one. So 1, 2. And A, B. A, B, C, D. So 1B, 2C, 3A. Then fourth one is D. 1B, 2C, 3D. Fourth one is A. So the answer is third one. Next is the question. Slime molds are saprophytic protista. All the slime molds are saprophytic protista. The body moves along with the decaying beak and leaves that engulfing organic material, engulfing the material under the suitable condition. They form a aggregation is called plasmodium. Plasmodium. These are the characteristics of animals and which may be spread over the several feet. So the correct option is fourth one. These are called saprophytic or decomposer protista, slime mold, and they form plasmodiums. And see the next question. Majority of the euclinoids are freshwater organisms found in the stagnant water, correct? It's not that cell wall, they have protein-rich layer is called pellicle. Pellicle is present, proteinous layer, which make their body flexible. Okay, they have dash flagella, 2 flagella. One is a long flagella, and one is a short flagella. So the correct option is A and B are respectively. First one A, B, A pellicle, okay. B 1 not true. Second, B pellicle. B pellicle no. And the third one is C gelatinous sheath. C no, it is a gelatinous no. Fourth one is B pellicle. No, it is a option. It's a B pellicle. And 2. B1 it is a 2 flagella. The correct option is fourth one. Next. See the question. The organism which are found in fresh water as well as marine environment, they are microscopic and a passive one. Microscopic passively in water current, that is called plankton. Most of them are photosynthetic. Cell wall forms two thin overlapping shell which are fit together in a soap box like structures. This is example for diatom. So diatoms which are belongs to the class chrysophytes also correct. Diatom also correct. The correct option is 2 and 3. The next question. Read the statement A to D. A is golden algae are classified under chrysophytes. It's correct. Golden brown algae classified under the chrysophytes. It's also called basidiophytes. For example, diatoms and desmites. Second option, goniolex and diatoms are classified under dinoflagellate. No. Diatoms are classified, goniolex are classified under dinoflagellate. Diatom no. Diatoms are classified under chrysophytes. Third option, in diatoms, the cell wall form thin overlapping shell which are fit together a soap-like pus, look like a soap-like pus. It's a correct one. Diatoms. Fourth one, most of the dinoflagellates have 2 flagella. This is also correct. So, how many statements are wrong statement? Only one. First one is a correct. So, the B one is a wrong statement. See the next question. Read the following 4 statement A to D and answer asked next to the dump. Next to dump. See the first option. All single-celled eukaryotes are placed under protista. Protista prokaryotes. It's a wrong. Single-celled prokaryotes are placed under. Sorry, all the single-celled eukaryotes. Yes, is correct. The single-celled eukaryotes are placed under protista. Correct. Second option. Most of the organisms of chrysophytes are photosynthetic. It is also correct because they are called photosynthetic protista. Third option. Dinoflagellates appears yellow, green, brown, blue, or red, depending upon the main pigment which is present in the cells. Yes, depending upon the main pigment, for example, chlorophyll A, chlorophyll C, fucoxanthin, diatoxanthin, diadenoxanthin. These are the pigments which are different colors. That is why they are called golden brown algae dinoflagellate. This is also correct. And the chloroplast absence in Euglena is wrong because certain plant characters also possess in Euglena due to the presence of chloroplast. So it is wrong. How many of the above statements are correct statements? First 1, 2, 3. 3 statements. So the correct answer is 2. Next question. Compared to slime mold, euglenoid has presence of chloroplast. Yes, correct. Holophytic nutrients. Yes, it is a photosynthetic or autotrophic nutrients. Presence of proteus pellicle is present. That is also correct. Presence of contracting items also present. Yes, it is also correct. So the option is A, B, C, D. All are correct. So because of presence of these characteristic features, euglenids are different from the slime ones. See the question number 121. Euglena is considered as a connecting link between plant kingdom and animal kingdom because there's a features of both plants and animals. Which are the following are plant characters of Euglena? Presence of chlorophyll pigment, yes, it is a plant characteristics of Euglena. Holozoic nutrients, it is an absence. Holozoic in the sense ingestions of food material, so the further digestion takes place within the body. These are called holozoic nutrient, but actually Euglena is a holophytic and absorbing types. When in the absence of light energy, they get their food material from decay and decomposing organic matters. So these are the absorbing types, not in holozoic. Holophytic nutrients present and a process of sexual reproduction. Sexual reproduction absent. So which is considered as a plant and animal characters A and C. A and C. So fourth option is correct. Next question number 122. Select the wrong tab. Red dye is caused by Gonialax. Yes, correct. Gonialax and the gymnodium that cause red dye. Cellulosic cell wall Archibacteria. No, yes, no. Because Archibacteria, most primitive bacteria, the cell wall is made up of non-cellulosic cell wall. That is nothing but muco. Murin is present. Sorry, pseudomurin is present. That's a wrong. And saprophytic protista slime mode is a correct one, correctly matched or bad. Mycoplasma cell-wall-less organism, correct. They can survive without oxygen. It's a correct. So the wrong pair is second one. Kingdom fungi is next unit. Yes, great. Yes. So the questions. Question number 31. In the five kingdom system of classification, which one is the main basis of classifications? I already point out while I am teaching. The five criteria are followed by R.H. Butagar to propose five kingdom system of classifications. Which one is the main mode of nutrients? These are the main criteria followed by him to classify the organism. Mode of nutrient is the main basis of classification. Next, in the five kingdom classification, the kingdom that includes blue-green algae, nitrogen fixing bacteria, and methanogenic archibacteria yeast is nothing but Monera. Monera, which includes archibacteria, eubacteria, blue-green algae is nothing but cyanobacteria, and mycoplasma. So all unicellular and prokaryotes comes under the kingdom Monera. Question number 33. Which statement is wrong statement? Both ascospores and basiospores are sexual spores formed endogenously. It's a wrong fungi. Ascospores are formed exogenously. Each ascus to produce 8 ascospores after meiosis. Ascospores. That's correct. But basiospores are formed Sorry, ascospores are formed endogenously. Ascospores formed endogenously inside the ascus after meiosis, but basilospores are formed exogenously. It's a wrong to produce four basilospores. And the second option, phonetic classification is based on the observable characters of organism. Yes, it's correct. Phonetic classification is nothing but numerical classification, which is based on only the morphological characters. To analyze the classification, to analyze the data based on their only morphological characters, so it is only observable characters of organism. Phonetic, it is also called Adansonian classification. So it is correct. Third option, monograph contain information of any one taxon. It is also correct. So monograph, it is one of the teaching aids, taxonomic aids, not teaching aids, sorry, taxonomic aids like key, monograph, and museum, zoological park. So these are the taxonomic aids which is used to identification of organisms based on their real specimens collected from different parts. So the monograph, it is one of the taxonomic aids which contain the information of any one taxon. It is also correct. And next, herbarium, it is a storehouses of collection of plant species and specimen that are dried, preserved, and preserved on the sheet. It is also correct. And which statement is a wrong statement? The first one is a wrong statement. See the question number 34. In the 5 kingdom classifications, Chlamydomonas and Chlorella have been included under Protista. But in early system of classification, Chlamydomonas and Chlorella kept under algae that belongs to Plantae, kingdom Plantae. But in RH Botany system of classification, Chlamydomonas and Chlorella, because these two are unicellular and eukaryotic organisms, true nucleus is present. That is why it is kept under the Protista. Question number 35. According to 5 kingdom system of classification of Botany, saprophytic slime are placed under kingdom Protista. Kingdom Protista. In Brassica oleracea capitata, it is a binomial of cabbage, Brassica oleracea capitata. The term capitata that refers to varieties. Variety name. First name refers to genus, second name refers to species name, third it is a variety names. Next, in taxonomic hierarchy, the less common features from the order will present in. The taxonomic hierarchy is 7 segment. The first one is kingdom, the second division, third class, then order, family, genus, species. If you take the ascending order, the similarities, species and organism similarities, it's gradually decreasing. Take the ascending order. And if you take the descending orders from kingdom to species, the similarities shows more common features are increasing. And what is the question? The taxonomic hierarchy, less common features from the order will present in class. From the order, the less common features class division of the kingdom also. From the order, the more common features are more similarities present family, genus, species. So from the order, less class. This is a correct point. Okay, dear students, and please go through that. And if you have any doubt regarding subject, And as usual, contact me through WhatsApp number and put your maximum effort towards the preparations. And your preparation is always towards your goal or your destinations. And thank you.